Hey guys, we're solving this bio paper today. This is 9700 variant 13, October November 2021. Let's begin. Let's talk about the threshold. Wait a second. So A was at 26, B was at 22, C was at 18, D was at 15, and E was at 12. So that's very low comparatively. So A star was probably at around 30 to 32. That's what you needed to get an A star for this paper. So you can test yourself. If you get that mark, you're good enough, okay? But it's always good to aim around 34 to 35 just to be safe, okay? So let's begin. Starting with question 1. A student was asked to use the scale bar shown to calculate the magnification of a cell on a photomicrograph. Which method could the student use to calculate the magnification of the cell? Okay, so we know that magnification is equal to image by actual. Divide the diameter of the cell by the length of the scale bar, with both measured in the same units of length. Measure the diameter of the cell in millimeters, multiply by 2000 and divide by the length of the scale bar measured in millimeters. Measure the length of the scale bar in millimeters, convert to micrometers and divide by 2. Measure the length of the scale bar in millimeters, convert to micrometers and multiply by 2. Okay, so magnification is equal to image length by actual length, right? So we need to understand something. What's the best answer here? The answer is actually C. I'll tell you why. So we're going to use a scale, right? We're going to use a scale to measure the length of this bar in millimeters, okay? Then we are going to convert that to micrometers. So that's the image length, right? And this 2 micrometers is actually the actual length. Understood? So since both of them have the same units, we don't need to convert it anymore, right? And then magnification is equal to image length, the one we just found out using the scale, converted it to micrometers, then divide by 2 to get, the magnification. That's it. So clearly D is wrong because multiplication is involved. We need to divide it. And the other ones, we aren't supposed to find out the diameter of the cell. Okay, that's a wrong method. Remember that. For 2, which statements about light microscopy are always correct? The greater the resolution, the greater the detail that can be seen. I agree with this 100%. This is right. The greater the magnification, the greater the detail. No. With magnification, you also require resolution. So 2 is wrong. And we just figured out that 1 is correct, right? So it has to be B. Increasing the magnification up to its limit of resolution allows more detail to be seen. Now this is good. This makes a lot of sense, right? The shorter the wavelength of light used, the greater the detail, right? The shorter the wavelength, the greater the resolution. Okay, this is also true. So 1, 3, and 4 are true. Number 3, what is the length of a typical prokaryote such as E. coli, Escherichia coli? So it's going to be C. It's around 1.5 micrometers. This is a bacterium. Okay, so what's the typical length of a bacterium? 1.5 micrometer works. Okay, like this would have been too much. What's the typical length of a bacterium if you guys want to know? It's about 5 to 10. 5 to 10, it's around 5 to 10 micrometers, right? And for a eukaryote, this is for a prokaryote. For a eukaryote, it's like 10 to 100 micrometers, okay? So 1.5 is the best one since it's okay. I mean, it's fine, right? Because if you look at these, what about these? These are at, this is 150 nanometers or 0.15 micrometers. That's too small. 0.15 is too small. This is 15 micrometers, right? So 15 is a bit bigger than 10. So 1.5 is, yeah, it's the better one. Think of it as 1 to 10, all right? Prokaryotes should be around 1 to 10 micrometers typically. Moving on to 4. 4 should be what are present in Plasmodium. So remember, guys, prokaryotes do not have. So prokaryotes do not have membranous organelles. But remember, this was a trick question. Plasmodium is actually a eukaryote. It falls under the domain Eukarya, but the kingdom is actually Protoctista. It is a protoctist, which is a eukaryote. So it actually has all three ribosomes, mitochondria, and Golgi body. Okay, you'll learn about this more in A2. Plasmodium fall under the kingdom Protoctista, and they are in the domain Eukarya. Which cell structures may contain a nucleic acid? Important question. The answer to this should be 1, 2, and 3. Ribosome contains rRNA. Chloroplast contains everything, DNA, RNA, everything. Okay. Cytoplasm also contains tRNA, and indirectly it contains the nucleus. So Golgi body does not contain nucleic acid. Which row shows correct features of ATP? The carbohydrate in ATP. Cytoproduction of ATP. Okay, this is an important question. ATP is produced in both mitochondria and chloroplast. Remember that. Both of them have the electron transport chain. This is the first thing you'll learn soon in A2. Now, what about the sugar? Is it deoxyribose or pentose to be exact? Adenosine triphosphate. It's a pentose sugar, right? Adenosine triphosphate. It contains the nitrogenous base adenine, a ribose sugar, and 3 ATPs. So remember, it's not a deoxyribose sugar, it's a ribose sugar. Okay. 7. Which tests will identify biological molecules that contain monomers with a carboxyl group. So this question was discontinued. I'm seeing the mark scheme. It was discontinued. Let me just see the examiner's report. Paper 1-3, question number 7, right? It was discontinued. So I'm not going to solve this actually since it was discontinued. Basically, this is for reducing sugars, right? And this is for proteins, right? And what is ethanol used for? It's used for the emulsion test for fats. So basically, they made a mistake in the question. Let's not solve 7. There's no answer for that. 8. Which polysaccharide is correctly described? Amylopectin does not contain beta glucose. Amylose contains alpha glucose, right? And it is used for storage. That's true. B is correct. Which formula represents a saturated fatty acid? Okay, which formula contains or represents, sorry, a saturated fatty acid? So let's try to understand this. Look at this. CH3, CH2, CH2, COH. CH3, CH2, 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 COH. If you look at the molecular formula, what you're going to see is this is actually C4, H3, 4, 5, 6, 7, 8, O2. This is C1, 2, 3, 4, 5, 3, 4, 5, 6, 7, 8, 9, 10, H10, O2. Try to see the trend. For a saturated fatty acid, do you see the ratio of carbon to hydrogen? What is it? Isn't it 1 is to 2? Do you see that? So out of all these four, where do you see a 1 is to 2 ratio? 18 is to 36. That's 1 is to 2, right? That's why A is the answer. In the other ones, you won't see a 1 is to 2 ratio. Those are unsaturated fatty acids, okay? We've seen a similar question in Feb March 2022. This is really important. Which molecules always contain at least 4 double bonds? We need 4 double bonds. Okay, at least 4. So the triglyceride, it contains 3 ester bonds. So there are 3 double bonds there, right? But how are we sure that there's going to be a fourth one? In the triglyceride, there are 3 for sure, but the fourth one depends on the nature of the fatty acids. So we can't say for sure. We can't. So basically, triglycerides are cancelled. Okay, it has to be C. Right? 10 should be C. Because why hemoglobin and collagen? They're proteins, right? You have multiple peptide bonds, so you're gonna get multiple double bonds from the CONH. Easy. Moving on. Which way about the structure of proteins is correct? The number of the primary structure is not the number of amino acids. This is a misconception, okay? The order of amino acids present, true. The coiling of the chain of amino acids to form beta plate sheet, okay? The shape formed by folding of a polypeptide and held together by hydrogen bonds only. It's held together by other bonds as well. So B is not the best one. The result of translation of an mRNA molecule by a ribosome into a chain of amino acids occurs between attraction between hydrogen and oxygen atoms in side chains. It's not from the side chains. Side chains refer to tertiary structures only. Okay, so not C. The sequence of amino acids true formed by hydrogen bonding between amino acids forming the primary structure. This is true, not the side chains. The CONH from the amino acids formed a result of interaction of side chains. Tertiary goes with side chains, right? 11 is D, right? 12. Which statement about collagen is correct? A collagen fiber is made of 3 parallel helices with hydrogen bonds holding them in place. It is an insoluble fibrous protein with a quaternary structure. 1/3 of the amino acids are not valine, it's actually glycine. Okay, that's wrong. Collagen fibers are formed from several collagen molecules held together by ionic bonds. That's wrong. It's actually not ionic, hydrogen. Okay, so it's between A and B. Which one is the better one? B is just the better answer. It is an insoluble fibrous protein with a quaternary structure. So what's wrong with A actually? If you guys are wondering, what's wrong with A? A collagen fiber is made up of 3 parallel helices. Let's see what's wrong. See this. This is from chapter 2, right? So if you go to collagen, it's probably here, right? Three helices bind together to form a collagen molecule. And many of these helices, 3 helices, lie side by side linked to each other by covalent crosslinks between the side chains of amino acids near the ends of polypeptides. Okay. To form. Okay, so 3 polypeptide chains, like the one shown in A, make up a collagen molecule. Many collagen molecules make up a fibril, and many fibrils make up a fiber. Okay, so that's different. And the most common amino acid is actually glycine. Okay, the 3 strands are held together by hydrogen bonds. Remember that. And there are covalent crosslinks between the side chains of amino acids near the ends of the polypeptides for like 3 helices to another 3 helices, something like that. Okay, remember that. Important. So it should have been a collagen molecule is made of 3 parallel helices. Okay. Now 13. Which properties of water are a result of hydrogen bonding? Solvent action, specific heat capacity, latent heat of vaporization. 2 and 3. The solvent action is not due to hydrogen bonding. It's just polar. Okay. The polarity makes it a good solvent for polar compounds. It's not. This was not in the book, and this is not. As a result of hydrogen bonding. Okay. Which descriptions about all enzymes are correct? All of them catalyze the breakdown of large molecules. Some of them may also catalyze, anabolic reactions. Okay, so this is wrong. Okay, one is wrong. Only function inside cells. No, there are some extracellular enzymes as well. So 3 and 4 are correct. 14 is D. 15. The cells in the roots of beetroot plant contain a red pigment. When pieces of root tissue are soaked in cold water, some of the red pigment leaks out of the cells into the water. An experiment was carried out to investigate the effect of temperature on the loss of red pigment from the root cells. It was found that the higher the temperature, the higher the rate of loss of red pigment from the root cells. Which statements could explain this trend? Enzymes in the cells denature as the temperature increases, so the pigment can no longer be used for reactions inside the cells and diffuses out. That literally makes no sense. Like, one is clearly wrong. This doesn't make any sense. As the temperature increases, the tertiary structure of protein molecules in the cell's surface membrane changes, increasing the permeability. This is true. Phospholipid molecules gain kinetic energy as the temperature rises, increasing the fluidity of the membrane and allowing pigment molecules to diffuse out more easily. Now that's true. 2 and 3 are in fact correct, so 15 should logically be B. Okay? The diagram shows a partially plasmolized plant cell X. What is found at Z? So you guys need to understand that this is the cell membrane and this is the cell wall. So what's found in Z, this region? Basically, this solution Y enters this region. So it should be solution Y. 16C, common question. 17. A single-celled organism lives in fresh water. Water that enters the cytoplasm of the cell by osmosis is collected into a structure called the contractile vacuole. To remove the water, the contractile vacuole fuses with the cell surface membrane. A student counted the number of times that the contractile vacuole filled and emptied when the cell was placed in solutions with different water potentials. The results are shown in the table. Which statement explains the pattern observed as the water potential of the external solution decreased? Let's see. The water potential gradient between the cell and solution increased, causing water to move into the cell more rapidly and the contractile vacuole to empty more frequently. Not really. As we decrease the water potential, the contractile vacuole actually empties less frequently. A is wrong. The water potential gradient between the cell and solution increased? No, it's not solution increased. Clearly, the difference decreased. That's why it did not empty anymore. At this point, the potential of that, cell and the solution actually becomes the same. That's why it doesn't empty. C. The water potential gradient between the cell and the solution decreased, causing water to move into the cell more rapidly and more frequently. No, it should be less frequently, and as it decreased, causing water to move. Yeah, this is correct. The gradient decreased and less frequently. D is correct. 17D. 18. Which row? Do you get it? Do you get 17? Basically, As we go down the group, it's becoming less frequent for sure. That's why these more frequent ones are cancelled out. And as we decrease the water potential, the difference between the cell and solution clearly decreased. That's why the rate of emptying also decreased. 18. Which row shows the correct number of each of components of a single chromatid during anaphase of mitosis? During anaphase, the chromatids basically move to this side. So each chromatid basically has, they're talking of about a single chromatid. Each chromatid has two telomeres, right? So two telomeres, and each of them has one centromere in the middle. So 18 should be A. And what about DNA molecules? There's only one DNA molecule, but two DNA polynucleotide strands. Okay, really tricky, but you should understand these. 19. Some processes are listed. How many processes occur during tissue repair by stem cells? Okay, so for tissue repair, we mainly need mitosis. Okay, so as you can see, for repair, we need totipotent or pluripotent, basically stem cells to repair it. We need cells that can differentiate into um, like stem cells will differentiate into a certain type of cell, suppose maybe roots or something. Okay, so of course we need DNA replication, mitosis and cytokinesis, and we also need differentiation for the stem cell to turn into the type of cell that's required. So we need all four processes. Okay, which stage of mitosis is not shown? This is actually prophase, this is anaphase, this is telophase, and this is actually after cytokinesis has taken place. So we aren't being shown metaphase. Metaphase is missing. 20C. 21. Which letter in the key describes uracil? Okay, so guys, short lecture on bases. They are either purines or pyrimidines, right? So purines are actually, the name is shorter, but they're double ring structures. Okay, that's adenine and guanine. Okay, what about pyrimidines? We have thymine, we have cytosine, single ring structures. We also have uracil. Okay, so uracil is not a pyrene, sorry. It has a single ring structure. Yes, so it should be C. 21 is C. Okay, now when a gene for protease is activated, which nucleic acid will be formed? So basically from DNA we transcribe DNA to form mRNA. Very important. Okay, the diagram shows a vascular bundle from the stem of a plant. Clearly, guys, one, look at the thickened cell wall. This is clearly the lignin, right? This is your xylem. One is basically xylem, and it transports mineral and ions, okay? So, I'm looking at these. I'm looking at B and D, okay? B and C, sorry. Yeah, I'm looking at B and D. Now, what next? Stores starch and transport sucrose. So, who actually transfers sucrose? Phloem, right? Clearly. So look at this. These are the phloems. This was the vascular bundle mainly, right? So this should be, two should be phloem, all right? So the B should be the answer. And stores starch. This is basically the parenchyma, okay? So you know it's the leaf cells, the parenchyma cells from the leaf. That's it. This is the stem of the plant, right? So not leaves, just parenchyma. Some of the features present in the transport tissues of plants are listed. Which features are present in sieve tube elements? Okay, we do not have lignified walls in sieve tube elements. Okay, that's gone. A and B are excluded. It's between C and D. 2, 3, and 5, 2, 4, and 5. Only 3 or 4 is the determining factor. Okay, sieve tube elements. Remember, sieve tube elements do not have mitochondria. Okay, that's why C is wrong. D must be the correct answer. It has scanty cytoplasm. It does have chloroplast, and it does have plasmodesmata connecting it with the companion cells. Okay, but no mitochondria. Clear? 25. Which statement explains why transpiration is an inevitable consequence of gas exchange in plants? Really common MCQ. The answer should be D. Hydrolysis reactions are taking place in cells. This has no connection. ATP production is occurring. Wrong. Stomata must be open. Okay, stomata must be open. It's an inevitable consequence of gas exchange. Because without stomata being open, the plant cannot, you know, take in oxygen for take in carbon dioxide for photosynthesis and give out carbon dioxide, right? Also it cannot take in carbon dioxide for photosynthesis and give out oxygen. Also it cannot take in oxygen for respiration, right? So while stomata is open, stomata the right while stomata is open, a water vapor also leaves the leaf. That's it basically. 26. In an investigation, a leafy shoot was attached to a potometer and exposed to a variety of conditions. The time taken for the meniscus to move 50 millimeters along the capillary tubing was recorded for each set of conditions. Which row shows the slowest and fastest rate of uptake for investigation? Okay, fastest, slowest. Millimeter per second, right? So to travel 50 millimeter, it took 40 seconds. To travel 1 millimeter, how long it will take? 40 by 50. That gives us a value of 0.8 millimeters per second, right? And the next one, 50 millimeters in 166 seconds. So 166 by 50, that's actually 3.3, 1 millimeter in 3.3 seconds. Okay, so that's the slowest rate. 3.3 slowest, and the fastest rate is 0.8 according to this my calculation basically. But look, we aren't getting an option of 3.3 and 0.8, right? We don't get that. So what's wrong really? Let's try to figure this out. Which row shows the slowest and fastest rates of water uptake for the following investigation? What about the other ones? Let's do those. 125 by 50. That's actually time taken to move 50 millimeters. 125 seconds. The rate is in millimeters per second. Okay, yeah. So here we actually figured it out that it took 1 second to travel 0.8. Sorry, it took 0.8 seconds to travel 1 millimeter. But look at our unit. What unit do we need? We need to figure it out in 1 millimeter per second. You get it? It needs to be 1 second. We need to figure out the rate in per seconds, basically. So how are we gonna do that? So the logic is it's like this. We did it the opposite way, to be honest. Okay, sorry about that. I just did the other way around. Mainly, it should be like this. You take 40 seconds to travel 50 millimeters, right? So in 1 second, how many millimeters should be traveled? That's actually 1.25 or 1.3 millimeters per second. Okay, this is the fastest rate, 1.3. What about the slowest one? We take 166 seconds to travel 50 millimeters. So 50 by 166 is in 1 second we travel 0.30 millimeter. Okay, so that's A. 26 is A. 27. Proton pumps and co-transporters are used by plants when loading sucrose into a phloem safety element at a source. Which row is correct? Proton pump pumps protons into a companion cell, raising its pH. No, we actually pump it into the cell wall. Pumps protons out of a companion cell into its cell wall. This looks good. 27C looks good. The other descriptions are wrong. we actually pump it out from the companion cell into the cell wall of the companion cell. Okay, and then what happens, the co-transporter actually brings the protons back into the companion cell along with sucrose. Okay, 28. The statements list some of the events in the cardiac cycle. They are not in the correct order. Which statement describes the sixth of these events to occur in the cardiac cycle? Okay, ready? 28. Let's start. At first, the sinoatrial node will contract. Okay, then a wave of excitation sweeps across the atria. Do you understand? Then the atria will contract. Okay, now the atrioventricular node will receive the impulse, and delay it for a fraction of a second so that the atria can empty. Understood? Then the impulses will travel through the Purkinje tissue. Then it sweeps upwards from the base of the ventricles, and at last the ventricles will contract. So which one occurs? Which is the sixth to occur? That should actually be 5D. Okay, 29. Which tissue types are present in the walls of all blood vessels? Okay, in all blood vessels we obviously have, remember, only endothelial tissue, because in capillaries we only have endothelium, nothing else. We may have elastic tissue, smooth muscle tissue, in veins and arteries, but no, in capillary we only have endothelial tissue. Okay, it's a trick question. 30. The photomicrograph shows 3 white blood cells labeled X, Y, and Z. So Z is clearly, it's like when you see a rim of cytoplasm, it's clearly a lymphocyte. Okay, Z is a lymphocyte. And remember I taught you in variant 1.1, do you see lobes in X? That's a neutrophil. Understood? X is a neutrophil, right? 30 should be D. Okay, so remember, if you see lobes, you're gonna see that it's a neutrophil or a phagocyte. X is a neutrophil. Z, when you see a rim of cytoplasm, it's gonna be a lymphocyte. And when you see a kidney-shaped nucleus, remember that it's a monocyte. Okay, Y is a monocyte. Easy. Last section of the video, 31. When active tissues have high carbon dioxide concentrations, oxyhemoglobin needs to release oxygen to the tissues. How is the carbon dioxide transported away by the blood from the tissues? Okay, so maximum carbon dioxide is transported as bicarbonate ions. Okay, maximum. And then we have carbaminohemoglobin. Carboxyhemoglobin is carbon monoxide binded with hemoglobin, not carbon dioxide. So a lot of candidates get tricked, but remember, try to be the smart ones out there. It's gonna be carbaminohemoglobin and bicarbonate. Some of it is dissolved in blood as well, but very little parts. So 31 should be C. 32. Which effect does increasing carbon dioxide concentration have on hemoglobin? Basically, it causes the release of oxygen from hemoglobin. Okay, it is less efficient at taking up oxygen and more efficient at releasing oxygen. That should be our answer. Okay, 32 should be B. That's called the Bohr effect, right? It was like this, but afterwards it becomes like this. It shifts to the right. 33. The diagram shows a magnified section, a part of the lungs containing specialized tissue. Which is correct for structures labeled 1 to 6? I've labeled them at the top. See, 1 is actually the alveolus, the epithelium of alveolus, squamous epithelium. 2 is actually the capillary endothelium. Okay, the blood vessels. 3 is RBC. 4 is the alveolus itself. 5 is the blood plasma, and 6 is the alveolar macrophage. Okay, it's a special type of cell in the alveoli. So which contains high proportion of carbonic anhydrase? We know that carbonic anhydrase is present in RBC, so it should be 3. Bicarbonate in plasma 5, and lysosomes in 6 because it's a macrophage. Important, okay? Remember this, guys. 34. The table shows some parts of an animal's lungs that contain different cell types. Okay, which part of this animal's lungs clean inhaled air and which carry out gas exchange? Remember, gas exchange is carried out by alveoli and the respiratory bronchioles. Alveoli, basically. Okay, and respiratory bronchioles, the terminal section of bronchioles, not the whole part, only the terminal section. Remember that. So Typically, when you're given the option, just think of it as alveoli only. You're gonna think that bronchial cannot. Okay. And cleaning, who's in charge of cleaning? Basically any section that has cilia, right, for the sweeping action. Goblet cells help as well because they help to stick, they're sticky and help to trap dust and foreign particles. So 34, the best answer should actually be, it should only be alveoli. Okay, so the answer should be D. Best answer 34D. What helps to maintain a steep oxygen concentration gradient between the air in an alveolus and the blood? Breathing in brings in a supply of air with a relatively high concentration of oxygen to the alveolus. This is true. Blood flow brings blood with low concentration of oxygen. That's true. So we get a gradient. The relatively low concentration of CO2 in the alveolus results in carbon dioxide leaving the RBC, allowing hemoglobin to combine with oxygen. Perfect. A is the correct answer. 36. Goblet cells are found in the trachea. Which cell structures would be found extensively in a goblet cell? Okay. Golgi body is required for packing. Mitochondria is required for, energy. And ribosomes are required for protein, glycoprotein synthesis, right? Protein synthesis. So 36 should logically be A. We require all three. Okay, malaria. In a country where malaria has successfully been eliminated, an outbreak of malaria can occur years later. What could allow this later outbreak of malaria to occur? They become resistant to insecticides. Sounds good. Migration of population due to war, true. Malarial parasites become resistant to quinine. That's a drug used for treatment. So yeah, all three are correct. Okay, we don't have 39 by the way. 39 is a question on autoimmune diseases. These have been excluded from the syllabus. Which show about a person with leukemia only and about a person with measles only is correct. So guys, leukemia is a cancer. I don't think we have measles anymore, right, in our syllabus. But still, I'm going to show you. Leukemia is a cancer of blood cells. Okay. So as a result, the number of WBC in a person with leukemia only decreases. Okay. So it's either, oh, wait, let me check this again. White blood cell count in a person with measles only. So if you have measles, since it's an infection, okay, since it's an infection, the WBC count has to increase. Okay, so we're gonna get rid of A and C. It has to increase. Now, what about the other options? Bone marrow function in a person with leukemia only. WBC count in a person with leukemia only. Okay, about this. Basically, what happens in cancer? We have uncontrolled mitosis. The number of cells increases by a great margin and it is uncontrolled. So yeah, the white blood cell count in a person with leukemia only does cause an increase due to uncontrolled mitosis. I actually meant that the number of like competent cells actually decreases, but the number in general increases. Okay, so it should be D. Bone marrow function in a person with leukemia only, that also increase. Bone marrow, the function of bone marrow is to produce WBC, right? So it should increase as well. Last one, 40. Tetanus is a bacterial infection. The graph shows the blood antibody concentration of two people. The graph shows on day 1, day 0, person G was injected with antibodies to the tetanus toxin and H was injected with the vaccine. Okay, what could be the result if H and G were infected with the tetanus bacteria on day 20? Check this out. I drew the figure afterwards. So G was given artificial passive immunity. Do you understand? That means he or she does not have memory cells. So when he or she gets infected, it will be a primary response. So it takes time to take action, It takes time of about 12 days to take action, as you saw in H. However, what happened to H? He already knows what the bacteria is like. So if he or she does experience the bacteria again, the memory cells will be quick to respond and the secondary immune response will be much faster. So look at the options. Tetanus antibodies would not be produced in person G. That's wrong. It would be produced, but it would take some time. Antibody production would peak after day 32 in person G. That's true, because look at this. For person H, it took 12 days from day 0, right? After the vaccine was injected. So for person G, he or she actually was infected on day 20. So day 20 plus 12 is actually on the 32nd day. So that's true. Antibody concentration would stay constant in each person age. That's wrong. And a second antibody peak would occur in person age that would be lower than the first week. No, for the secondary immune response, remember the peak must be higher than the primary one. Okay, so that is it, guys. We're done with the October November 2021 series. If you like the content, remember to subscribe to the channel. Right, I'm gonna link paper 1 up here, and I'm gonna link ON 2021 variant 1.1 up here, and ON 2021 variant 1.3 down here. And next I'm gonna do the May June series for 2021 and March 5th 2021. Okay, so keep on supporting the channel. See you guys.